Hello friends, welcome back to BioOneEducation. In episode 3, we explored biomolecules. Today in episode 4, we zoom into surface of the cell membrane and discover how it decides what goes in and what stays out. Our topic is cell membrane and transport. We will cover the fluid mosaic model, passive and active transport, osmosis, and finish with the exam trick and animations. By the end, words like plasmolysis, facilitated diffusion, and endocytosis will feel easy. Let us begin. This chapter score well in NEET, 2 to 3 question most years. It also forms the foundation for plant-water relations in later chapters. So learning it will pays off twice. The good news, the chapter boils down to two big ideas. One, the membrane structure, which is fluid mosaic model. Two, the different ways molecule cross that membrane. Passive transport uses no energy. Active transport needs ATP. For each type, just to learn the direction of movement, the energy source, and one classic example. That is enough to answer almost any question they throw at you. The cell membrane is built on the fluid mosaic model proposed by Singer and Nicolson in 1972, and it is the currently accepted model. Why fluid? Because the phospholipid molecules are not stuck in one place. They slide laterally past each other, like people in a crowded train. The membrane flows. Why mosaic? Because protein are dotted across the bilayer in a scattered pattern, like tiles in a mosaic artwork. And the most important property, the membrane is selectively permeable. It does not let everything through. It picks and chooses which molecule can pass. That is why this chapter exists. The membrane is the cell security gate. Every membrane is built from one repeating unit, the phospholipid. Picture a lollipop with two sticks. The head is the round part made of phosphate group and glycerol. It is polar, hydrophilic, and water loving, so it faces outward toward the watery surroundings. The two tails are fatty acid chains, non-polar, hydrophobic, water-fearing. They tuck themselves inward away from water. When millions of these phospholipids arrange themselves, they naturally form a bilayer, two-layer arranged tail to tail. The hydrophobic core act like a security barrier, blocking most water-soluble molecule from passing through. And sitting between the phospholipid, you will find cholesterol molecule. Cholesterol acts as a fluidity buffer. It keeps the membrane not too rigid the cold and not too fluid in the heat. The membrane is not just phospholipids. Proteins do most of the work. Two types, integral or intrinsic protein, are embedded deep inside the bilayer. Some stretch all the way across from one side to other. These are called transmembrane protein. They form channels and carriers that let specific molecules through, and they act as receptor for signals. They are tightly embedded and cannot be easily removed. Peripheral or extrinsic proteins sit on the surface, either on the inside or the outside face of the membrane. They work as an enzyme, signaling relays, or structural anchors. They are loosely attached and can be washed off relatively easily. For the exam, remember the six major functions of membrane proteins. Protein transport. For the exam, remember the six major functions of membrane: protein transport, enzyme activity, signal transduction, cell-cell recognition via glycoproteins, intercellular joining through tight junctions, and attachment to the cytoskeleton inside the cell. Before we dive into each type, let us set up the big picture. There are two roads a molecule can take across the membrane. Passive transport is a free ride. Molecules move down their concentration gradient from high to low, like a ball rolling downhill. No energy needed. Diffusion, facilitated diffusion, and osmosis are all passive. Active transport is the uphill climb. Molecules are pushed against their concentration gradient from low to high, which takes energy in the form of ATP. The sodium potassium pump and bulk transport like endocytosis and exocytosis are active. Here is a simple exam shortcut. If the question mention against the gradient or ATP, the answer is active transport. If it is says down the gradient or no energy, it is passive. Let us start with the passive side. Simple diffusion is the simplest small non-polar molecule like oxygen and carbon dioxide just slip straight through the lipid bilayer from where they are more concentrated to where they are less concentrated. No protein help needed. But what about molecules that cannot cross the hydrophobic core on their own, like glucose or ions? They use facilitated diffusion. Protein helpers build into the membrane. Still passive, still no ATP, just assisted. Two kind of helpers. Channel proteins form a water-filled tunnel that a specific ion can pass through. Some of these channels are gated. They open and close in response to signals. Carrier proteins work differently. They grab the molecule, change shape, and release it on the other side. Carrier are slower than channels. The key exam point facilitated diffusion is still passive. No ATP, the protein just helps. The concentration gradient provides the driving force. Now, the star of this chapter osmosis. It is the net movement of water through a selectively permeable membrane from where water is more concentrated, the dilute side, to where it is less concentrated. Osmosis is really just diffusion of water. Three scenario every NEET student must know based on the solution outside the cell. Hypotonic solution. The outside is more dilute, so water rushes into the cell. A plant cell become turgid, firm and plump, which is good things for plant. But an animal cell which has no rigid wall can swell and burst. This is called lysis. Isotonic solution. Concentrations are equal on both sides. So there is no net movement of water. The cell stays the same size. Dynamic equilibrium. Hypertonic solution. The outside is more concentrated, so water leaves the cell. A plant cell undergoes plasmolysis. The membrane pulls away from the wall. An animal cell shrivels, that is called crenation. Here is osmosis in a single looping animation. Watch the blue water molecule on the dilute side cross the membrane toward the orange solute on the concentrated side until the distribution evens out and net movement stops. Play it a few times and narrate. Start, flow, balance, equilibrium. If you can do that without reading the labels, you have understood osmosis. Let us see osmosis in action inside plant cell. Because plant cells behave differently from animal cells thanks to their rigid wall. Turgidity in a turgidity in a hypotonic solution, water enters the cell, the vacuole swells, and the membrane pressure hard against the cell wall. The wall pushes back with turgor pressure. This firmness is what keeps plant upright. When your plant wilts, it has lost turgor pressure. Plasmolysis. In a hypertonic solution, water leaves the vacuole, shrinks, and the plasma membrane pulls away from the wall. The cell looks collapsed inside. The good news, plasmolysis is reversible if you put the cell back into the hypertonic solution. Imbibition. This is a different process. Dry hydrophilic material like seeds or dry wood absorb water by absorption, not through a semi-permeable membrane. That's why seeds swell before germination. And wall pressure. The rigid plant cell wall pushes back against the swelling cell, which is why plant cells don't burst in hypotonic solution, unlike animal cells which can lyse. Active transport. Here, molecules are pushed against their concentration gradient from low to high, which takes energy from ATP. The carrier protein involved are called pumps. They grab the molecule on one side, use ATP to change shape, and release the molecule on the other side. The famous example is the sodium potassium pump. It pumps three sodium ion out and two potassium ions in per cycle. This pump maintains the electrochemical gradient that your nerves and muscles depend on it. 4 quick facts for the exam. Direction is against the gradient. Energy comes from ATP hydrolysis. Carrier protein called pump do the work. And the classic example is the sodium potassium ATPase with its 3 out 2 in ratio. Here is the pump cycle in one continuous loop. Watch the molecule enter from the low concentration side, ATP attach and provide energy. The pump change shape and the molecule get pushed out to the higher concentration side against the gradient. The pump then reset and does it again thousands of times per second. This is active transport in action. For revision, narrate the 4 steps as they play. Bind, ATP, shape change, release. That one loop covers the whole mechanism. Bulk transport, endocytosis, exocytosis. What about really large molecules that cannot fit through any channel or pump? The cell uses bulk transport, wrapping them in pieces of membrane. Endocytosis is the way in. The membrane folds around the material and pinches off to form a vesicle inside the cell. Two types, phagocytosis, cell eating for solid particle like bacteria, and pinocytosis, cell drinking for liquid droplets. Exocytosis is the way out. A vesicle inside the cell fuses with the membrane and dumps its content outside. This is how your cells secrete hormones, enzymes, and neurotransmitters. Memory hook. Endo means enter, exo means exit, phago means eat, pheno means drink. And both endocytosis, exocytosis need ATP, so they count as active transport. Transport at a glance comparison table. Here is the whole transport chapter in one comparison table. Take a moment to scan it. This is your revision. Simple diffusion. I too low, no energy, no protein for small non-polar molecule like oxygen. Facilitated diffusion. I too low, no energy but uses channel or carrier proteins for glucose and ions. Osmosis. I too low, water only through aquaporins. Active transport. Low to high. ATP needed. Carrier pumps like the sodium potassium pump. Endocytosis bring big things in. ATP needed. Membrane wraps around. Exocytosis pushes big things out. ATP needed. Vesicle fuses with the membrane. Screenshot this table. It covers every transport type of exam can ask about. NEET memory tricks quick recall toolkit. Time to gather every trick in one card. Pause and screenshot this. Fluid means flow. The phospholipid move laterally 3 out 2 in the sodium potassium pump ratio per ATP. Endo means enter. Exo means exit. Hypotonic means the cell swells. Turgidity or lysis. Hypertonic means the cell shrink. Plasmolysis or Crenation. And if the question says no energy, the answer is passive transport. Say these 6 out loud a few times and they will be right there when the examiner tries to trick you. Real examples and current science. Let us step outside the textbook because these concepts are at work in real medicine and real Nobel Prizes. Peter Agre won the 2003 Nobel Prize for discovering aquaporins, special protein channels that let water cross membranes far faster than simple diffusion alone. They are the reason osmosis is so efficient in your kidney. Cystic fibrosis is a genetic disease caused by a single defective chloride ion channel called CFTR. One faulty channel protein leads to thick, sticky mucus in the lungs. It is perfect example of how membrane transport directly impact health. Scientists now design liposomes, tiny artificial phospholipid vesicles to carry drugs straight into target cells. They are using a membrane's own language to deliver medicine. And the dialysis machine mimic the selective permeability of real membrane to filter waste from blood when the kidney fails. Osmosis and diffusion save lives in hospital every single day. Surprise round, where NCERT hides lines that become direct questions. In membrane, the fluid mosaic model is the accepted model. Osmosis is a special case of diffusion. Cholesterol stabilizes membrane fluidity, and plasmolysis is reversible if the cell is placed back in hypotonic solution. In transport, phagocytosis means cell eating. The sodium potassium pump moves 3 sodium out and 2 potassium in. Aquaporins are dedicated water channels, and imbibition does not require semi-permeable membrane. More hidden gems. Facilitated diffusion is still passive despite needing proteins. Active transport uses carrier proteins, not channel protein. Turgor pressure keeps plant erect, and exocytosis is essentially the reverse of endocytosis. NEET asked this chapter in 4 costumes. Let us unmask them. Assertion and reason. For example, assertion: the membrane is selectively permeable. Reason: it has a phospholipid bilayer with embedded proteins. Both true, and the reason correctly explain the assertion. Matching the column. Pairing processes with their description. Phagocytosis with cell eating, osmosis with water diffusion, sodium potassium pump with active transport. How many statements are correct? Usually mixing up passive and active or confusing hypotonic and hypertonic direction. Read every statement carefully. Diagram based, they show a sketch and ask you identify plasmolysis versus turgidity. Label the bilayer or distinguish endocytosis from exocytosis by the direction of the arrow. Rapid fire round. Pause the video, answer in your head, then play again to check. If you got 4 or more right, you are exam ready for this chapter. If not, rewatch the osmosis and transport animation once more, and you will own it. Let us wrap up. Thank you for studying with me today. I will see you in the next one.